Okay, this is the first video on pathogens of human beings. There are many infectious agents that cause disease in humans. There is no way we could do all of them justice in just a few short lectures. There are entire courses devoted to infectious disease, and I encourage you to take some of them in Madison if you're interested. The diseases I have chosen, I did with purpose. All of them cause significant distress to the human population. Some of them, shown on the left, will be highlighted because of the important concepts they illustrate. Others are covered just because they are important. So what you need to know. For each disease, I want you to know the cause of the illness. I want you to know the general properties of the microbe that causes the illness. Be able to describe the course of the disease. Know the mode of transmission. And then something about the pathogenesis. For example, be able to explain the virulence factors the pathogen uses to compromise its host and survive. And then how it's treated. Escherichia coli is the first organism that we're going to look at. It has an interesting relationship with humans. It's mainly a commensal or mutualistic organism, even synthesizing B12 and vitamin K for its host. It also has been of great utility in scientific research. Yet, when certain genetic components are added to it, it becomes a significant pathogen. You probably already know this, but just in case you don't, E. coli is a gram-negative, non-spore-forming, short rod to ovoid shaped cell. And there's a picture up there at the right. Its optimal growth is at 37 degrees centigrade, facultative anaerobe, it's metabolically capable. It can ferment lactose to acid, and it's capable of forming indole. Both of those are distinguishing characteristics of the organism that can help to identify it. Further serotyping can be done, and it often is, to differentiate strains. Serotyping is basically using antibodies that react with different parts of the organism. There will be antibodies that react with the O antigen, which is the LPS, the K antigen, which is the capsule, and the H antigen, which is the flagella. These will then sometimes be used to type the strain. So O157H7, E. coli O157H7 is a very famous strain that causes significant illness, and that is being serotyped. There's also different pathivores, and these are organisms that have certain virulence factors in their genome. They have a certain set of factors in their genome that makes them able to cause a disease. There are 3 general types of infection and 9 pathivores. Number 1, there are urinary tract infections. Number 2, there are strains of E. coli that cause sepsis or meningitis. And finally, there's enteric diarrheal diseases that have been separated into 7 different pathivores, and they're listed here. I won't go into all of them. The one we're going to focus on is enterohemorrhagic E. coli, EHEC, but we'll do that in a second. So first of all, I want to say a little bit about colonization. All of these E. coli strains that colonize the intestines, the ones that cause gastrointestinal infections, have fimbriae, and these enhance their intestine colonizing ability. They allow adherence to the small bowel mucosa. Normally, commensal E. coli will live in the colon, not the small intestine. The different pathogenic strategies are due to differences in the genetic background. They'll either have unique plasmids, unique pathogeny islands, or they will code for proteins in other places that cause the specific illness. EHEC is probably the most significant E. coli infection. It's also called STAC, or Shigatoxin E. coli. There are about 73,000 cases each year in the United States. The incidence of EHEC infection has been rising. This is partly due to large food processing plants, especially for ground meat, increasing in their prevalence. And this makes it more and more likely that you'll get these kind of infections. More recent cases have been linked to vegetables and raw milk products that have been contaminated with cattle feces. Clinical manifestations of the disease are the following. EHEC is transmitted by food and water, but also by person-to-person contact. It has a very low infective dose of only 100 to 200 microbes. It's the most important source is cattle, but can be found in a wide variety of animals. Contamination of ground beef is common. Also, mayonnaise, unpasteurized apple juice, hard salami, raw vegetables, and water have been sources of this illness. Most outbreaks are caused by O157H7 serotype, but 50 serotypes have been known to cause the disease. This disease, the illness, was not recognized until 1982. The incubation period is about 3 to 4 days. It can be as long as 8 or as short as 1. The symptoms include abdominal pain and a short-lived fever, followed by non-bloody diarrhea. Vomiting occurs in about 50% of patients at this time. Within 1 to 2 days, the diarrhea turns bloody with increased abdominal pain, and this stage lasts 4 to 10 days. This is awful. I got a chance to talk to a former student who actually had it, and they said it was horrible. You feel terrible. In most cases, the disease then subsides without further complications. However, in about 10% of patients, there can be a complication called hemolytic uremic syndrome, or HUS. This will happen in the very young, less than 5, and those older than 65. The toxin produced by the microbe escapes into the bloodstream, and there it begins to kill erythrocytes. This causes them to accumulate in blood vessels and cause damage. It can also cause damage to kidneys and kidney failure. In both cases, you get high blood pressure just basically because of the clogging of the blood vessels. About 4% of children with hemolytic uremic syndrome die, and another 12 to 30% have serious complications. So, what are the things that this E. coli has that make it so nasty? We're going to talk about 2 of them. First of all, it has this ability to attach and efface, so it sticks very tightly to the cell, and the small intestine is difficult to get rid of. It will first initially attach through fimbriae, and then it will use a type 3 secretion system to inject its own receptor. It will pass through a translocated intimin receptor. This receptor goes in, it goes to the host membrane, and then intimin, a protein on the surface of E. coli, binds to the tear. So it brings its own receptor, sends it into the host cell, and then binds to it. It, of course, secretes a bunch of other host factors into the cell that cause a number of different problems. And then one of the things it does is it forms these pedestals. And you can actually see these in electron micrographs. Over here in A, you can see E. coli sitting on top of one of these pestles in an intestinal mucosa. Here is a slice, a transmission electron micrograph showing E. coli sitting on one of these pedestals. And these pedestals bind it very tightly, so it's very difficult for the immune system to get rid of this organism once it's in the small intestines. The other major virulence factor that it has is Shiga toxin. And this is a toxin that's encoded on a lambdoid phage that's lysogenic and can integrate into the E. coli genome. It's called Shiga toxin because this toxin was first discovered in Shigella. However, since this is a Shigella toxin encoded on a phage, the expression or secretion system that's used to export the toxin out of the cell is not present in E. coli, so there's no mechanism for secretion from the cell. Phage induction induces expression of the toxin, and then phage-mediated lysis of the bacterium releases the toxin. Shiga toxin is a classic AB toxin. The B subunit binds host global triacyl ceramide glycolipids, or GB3. These are abundant in the intestines and kidneys of humans. Interestingly, cattle lack these receptors. So they can have this organism in their intestines and it causes no disease. The A subunit is an enzyme that targets the ribosome and catalyzes the cleavage of ribosomal RNA. The toxin rapidly inactivates the ribosomes of the cells, thus killing the cell. There are also other effects of Shiga toxin. They include a stimulus of pro-inflammatory cytokines by a number of different host cell types. including the epithelium, so it increases inflammation, and as a pro-apoptotic stimulus, and that encourages the death of the cells that it infects. So this is a very toxic protein. Diagnosis and treatment. Diagnosis is by culturing the organism, and there are serological tests against the LPS and flagella to identify the strain. There are other more rapid tests. There's a test for the 015 antigen that's available, and you can detect the microbe directly in stools in less than 1 hour. Toxin-producing strains of the toxin itself can also be detected in stools using serological assays. And finally, rapid detection can lessen the chances of progression to hemolytic uremic syndrome, because once you know that an individual has this, steps can be taken to prevent the occurrence of pus. Treatment is largely limited to supportive care. You do not want to give the individual antibiotics. If you do, that will kill E. coli, it will induce the phage, and in that case, more Shiga toxin will be produced. Okay, clicker questions. E. coli initially attaches to the small intestine using... Okay, second question. Shiga toxin works by... All right, the answer to the first question is that E. coli initially attaches to the small intestine using Fimbriae. Answer to the second question is Shiga toxin works by digesting the ribosomal RNA of ribosomes. Okay, that's it for E. coli.